I was reading this quote from the astrophysicist Neil deGrasse Tyson, and it completely, it just shatters how you see your place in the universe.
Oh, yeah. Which one?
Well, he pointed out this thing about biology, and I'm quoting this directly from the source material. He said, within 1 linear centimeter of your lower colon, there lives and works more bacteria, about 100 billion, than all humans who have ever been born.
Right. It's a staggering number.
100 billion in 1 centimeter.
Mm.
And yet you and I walk around every day acting like, we are the ones in charge of the world. It actually pairs perfectly with this observation from Kurt Cobain. He once said, I sing and play the guitar, and I'm a walking, talking bacterial infection.
I mean, he wasn't wrong. It forces a massive perspective shift, really. We operate under this illusion that we are single autonomous beings, but biologically speaking, that is just not true. You are a walking ecosystem, like your continent.
Which brings us to the mission for today's deep dive. We are taking a comprehensive medical microbiology text, and we are going to map this hidden microscopic world that's basically inside and all around you.
Exactly.
We're going to decode how these tiny organisms dictate human health, the incredibly clever and honestly sometimes terrifying weapons they use, and fundamentally just how they operate.
Florence Nightingale actually captured the essence of why we need to do this. She argued that the wise and humane management of the patient is the best safeguard against infection. But to manage it, to actually safeguard yourself, you have to understand exactly what you are fighting. You have to know the enemy's blueprint.
Right, so let's get into that blueprint. Looking at how scientists classify all these billions of bacteria, it seems like everything starts with this primary laboratory test called a Gram stain.
Yes, the Gram stain is foundational.
And the sources describe it separating bacteria into two main camps based on color, right? Purple and pink. A lot of people compare it to a microscopic dress code. But reading through the actual mechanics of the cell walls, a dress code feels a bit too, arbitrary. It felt to me more like, okay, let's unpack this. It's like a sponge versus a raincoat.
I actually prefer that analogy. It gets much closer to the physical reality of what's happening. When a lab technician applies that initial purple crystal violet dye, the bacteria respond differently based on their physical armor. So the gram-positives have this massive, thick outer layer of something called peptidoglycan.
Is that the sponge?
Yes. Think of peptidoglycan as a thick, microscopic chain-link fence that basically acts like a sponge. It absorbs and holds tightly onto that purple dye.
Okay, got it.
But the gram-negatives, so the other camp, they have a very thin layer of peptidoglycan. And critically, they cover it up with a slippery outer lipid membrane. That's your raincoat.
Right, so the purple dye just washes right off them.
Exactly. The dye washes away, so they have to be dyed with a pink counterstain just so we can actually see them under the microscope.
But, if I'm lying in a hospital bed with an infection, why do I care if the bacteria attacking me is wearing a purple sponge or a pink raincoat? Like, what does that color actually tell a doctor?
Well, it tells them what kind of weapons the bacteria might be hiding. That pink raincoat, the outer membrane of a gram-negative bacteria, contains a molecule called endotoxin, specifically a component called lipid A.
The text describes endotoxin almost like a built-in grenade. But I was a little fuzzy on how a piece of the bacteria's own body acts as an explosive.
It comes down to what happens when the bacteria actually dies. We tend to think of bacterial death as them just quietly fading away, right?
Sure, like they just stop moving.
But they don't. When a gram-negative bacteria is killed, maybe by an antibiotic or your own immune system, it bursts open like a water balloon. We call that lysing.
Oh, wow.
And when it lyses, that lipid A grenade is ripped out of the membrane and basically goes off in your bloodstream.
So the danger isn't even just the living bacteria attacking you. It's the shrapnel from the dead ones.
Precisely. And your body completely overreacts to that shrapnel. Your immune system detects that lipid A and panics, triggering a massive release of inflammatory molecules.
Like what kind of molecules?
Things like TNF and interleukin-1. So for you, the patient, that immune overreaction translates to severe fever, life-threatening drops in blood pressure, and toxic shock.
That is wild. You can successfully kill the bacteria and still lose the patient to the endotoxin.
Unfortunately, yes. That's why managing gram-negative infections is so delicate.
Okay, but the armor isn't just about explosive raincoats, right? Let's talk about capsules. Some of these bacteria secrete these complex sugar coatings, polysaccharides, and just wrap themselves in them. The sources explicitly say this acts like an invisibility cloak against our immune system.
It's a very effective defense mechanism.
But I know our white blood cells normally cruise around eating invaders, a process called phagocytosis. Are you saying the white blood cells just can't see the bacteria if they have the sugar coating?
Well, it's less about visual invisibility and more about physical repulsion.
Mm,
Your white blood cells, let's call them the Pac-Men of your immune system, they want to engulf and destroy the bacteria.
right.
But that sugar capsule is highly charged and hydrophilic. It's incredibly slippery. The white blood cell tries to grab the bacteria and it just slides right off.
So how do we ever clear them out? I mean, if our standard immune cells can't even grab them, how do we survive an encapsulated infection?
This highlights a really crucial vulnerability in human anatomy. Because the normal white blood cells in your tissue can't handle them easily, we rely heavily on a specific organ to filter them out of the blood, the spleen.
The spleen.
Yes, your spleen contains specialized macrophages that basically tag these slippery bacteria for destruction, a process we call opsonization,
Wait, but there are people walking around without spleens. Like people get into car accidents and have to have their spleen removed all the time.
which puts them at massive immediate risk. Patients who are asplenic have a drastically lowered ability to clear these specific encapsulated bacteria.
Oh man, so what do they do?
If someone has their spleen removed, a doctor must immediately administer specific vaccines. Things like the pneumococcal, meningococcal, and Haemophilus influenza type B vaccines. You have to artificially give their immune system the highly specific tools to fight those invisibility cloaks, because they no longer have the organ designed to do it naturally.
That makes so much sense. Okay, so we're dealing with bacteria wearing explosive raincoats and slippery invisibility cloaks. But the sources point out they don't just evolve these traits in isolation, they share them.
They do. They are incredibly social in that way.
Yeah, they trade genetic secrets through horizontal gene transfer. And the first method listed is transformation. The text says this is when bacteria pick up naked DNA from dead cells in their environment. So wait, so transformation means they're basically scavenging superpowers off a microscopic battlefield.
I mean, it sounds like science fiction, but yes. Imagine a battlefield where a bacterium is killed, it lyses, and its DNA spills out into the environment.
It's floating there.
Exactly. A neighboring bacteria, one that is deemed competent, can physically bind to those floating discarded DNA fragments, pull them inside, and permanently incorporate them into its own genome.
That is insane.
It can literally pick up the genetic code for antibiotic resistance from the corpse of its neighbor.
That feels like a massive design flaw in nature from our perspective. But they also have more direct ways of sharing, right? Like conjugation. The notes mention a mating bridge.
Right. Conjugation requires physical contact. One bacterium extends a structure called a sexpulus. Think of it as a microscopic grappling hook that turns into a hollow tube.
Okay.
It connects to another bacterium and essentially downloads a copy of its plasmid DNA right across the bridge. Plasmids are these small circular pieces of DNA that often carry the blueprints for toxins or antibiotic resistance.
Like beaming a software update directly into another cell. But the one that really stopped me in my tracks was transposition, jumping genes. How does a gene just jump?
What's fascinating here is this is perhaps the most concerning mechanism when we talk about multi-drug resistant superbugs. Transposomes are specialized, highly mobile segments of DNA. They have the ability to literally copy and excise themselves from one location and insert themselves into an entirely different DNA molecule.
Wait, so a gene could jump from the main bacterial chromosome onto one of those mobile plasmids we just talked about.
Exactly. And once that resistance gene jumps onto a plasmid, that plasmid can be conjugated shared across the mating bridge, not just to nearby clones, but sometimes entirely across species lines.
You kid.
No, a harmless gut bacterium could pick up a resistance gene and just pass it to a deadly pathogen.
So they're actively upgrading their arsenals in real time, which I guess brings us to the actual weapons. We talked about endotoxins, the structural grenades, but then there are exotoxins. The reading says these are actively secreted polypeptide proteins, and the toxicity difference is staggering.
Oh, it really is night and day.
Yeah, like you measure a fatal dose of endotoxin in hundreds of micrograms. But exotoxins can be fatal in just one single microgram. Why is the lethal dose so infinitesimally small?
It comes down to precision. Endotoxins that just cause massive chaotic inflammation. Exotoxins are highly specialized proteins designed to target and dismantle specific cellular machinery.
Okay, so it's a sniper versus a grenade.
Great way to put it. They don't need to blow up the whole factory. They just quietly remove the one gear the factory needs to run.
Give me an example of that.
Take the Clostridium family of bacteria. Clostridium tetani produces an exotoxin called tetanospasmin. Once it enters your body, this toxin physically travels up your motor neurons right into your spinal cord.
Oh, wow.
And its specific job is to block the release of inhibitory neurotransmitters. specifically GABA and glycines.
Wait, if it blocks the inhibitors, the signals telling the muscles to relax get shut off.
Yes, your brain is trying to tell the muscle to rest, but the signal never arrives. The motor neurons fire continuously, without pause.
So the muscles just lock up entirely.
Exactly. That's the mechanism behind severe spastic paralysis, commonly known as lockjaw. The force of the muscle contractions can literally break the patient's own bones.
From a single microgram of protein, that is terrifying. Now, the notes contrast that with its cousin, Clostridium botulinum. It also produces a potent exotoxin, but this one does the exact opposite. It blocks the release of acetylcholine at the neuromuscular junction.
Right, and acetylcholine is the chemical messenger that tells a muscle to contract. If you block it, the muscle cannot fire, no matter how hard the brain tries.
So you get flaccid paralysis, complete limpness.
Yes, you see descending flaccid paralysis, which usually starts at the face and moves downward, eventually paralyzing the diaphragm so the patient stops breathing.
But hold on. Paralyzing the facial muscles by blocking acetylcholine, isn't that just Botox?
It is the exact same toxin. We take a highly purified, heavily diluted version of botulinum toxin and inject it intentionally to paralyze facial muscles and smooth out cosmetic wrinkles.
We are injecting one of the most potent, deadly bacterial exotoxins known to science directly into our foreheads for aesthetics.
We are. It highlights a strange truth about microbiology. Understanding the precise mechanism of a deadly weapon sometimes allows us to domesticate it.
All right, now that we understand their genetics and their neurotoxins, let's look at the actual armies on the ground. Let's start with the gram-positives, the purple sponges. Two major families kept popping up, Staphylococcus and Streptococcus.
Right. Two very common players.
They sound identical to a layperson, but the sources say they operate very differently.
Visually, under a microscope, they are both spherical. We call them coccia. But Staphylococcus aureus grows in these dense golden clusters that look like grapes. It is a formidable, highly aggressive pathogen.
And one of its most terrifying weapons is something called a superantigen, right?
Yeah.
Specifically, toxic shock syndrome, toxin 1. Superantigen sounds like something out of a comic book. What makes it super?
Normally, your immune cells are highly specific. An immune cell only reacts when it perfectly matches a specific piece of a specific invader. It's a lock and key mechanism, ensuring your immune system only deploys the exact force necessary.
Okay.
A superantigen completely circumvents that lock and key. It forcefully binds immune cells together from the outside, indiscriminately activating up to 20% of your body's entire T-cell population all at once.
Wait, 20%? So it's basically hitting the panic button for the entire immune system simultaneously.
Exactly. It triggers an overwhelming systemic release of inflammatory cytokines. The body basically attacks itself. This leads to toxic shock syndrome fever, rash, massive drops in blood pressure, and multi-organ failure.
Just from the immune system overreacting.
Yes. And that's not its only trick. Staph aureus also produces an enterotoxin that causes rapid onset food poisoning.
And the notes say that toxin is heat stable, which means if the bacteria gets into my food and pumps out that toxin, cooking the food might kill the bacteria, but the toxin survives the heat. I eat it and I'm still sick.
That's a critical distinction in food microbiology. Now, contrast staph with Streptococcus pyogenes, or group A strep. These don't grow in clusters, they grow in long chains. It's responsible for strep throat and scarlet fever. But its most deceptive tactic involves the immune system.
You're talking about molecular mimicry. I read this section and it sounded like biological identity theft.
It's a brilliant evolutionary disguise. If we connect this to the bigger picture, it's not always the bacteria that damages us, it's our own confused immune system. Strep pyogenes has a structure on its surface called an M protein, which helps it resist being eaten by white blood cells.
Right.
But the danger lies in its genetic code. The sequence of amino acids in that bacterial M protein looks structurally almost identical to human cardiac myosin.
Which is the protein that makes up your heart muscle.
Exactly.
Wait, if the bacteria looks like my heart tissue, what happens when my immune system builds antibodies to fight the bacteria?
Your immune system successfully builds antibodies that target and destroy the M protein on the strep bacteria. But because of that sequence homology, those same antibodies circulate through your blood, arrive at your heart, and mistake your own cardiac tissue for the bacteria.
So my own antibodies attack my heart valves because they're confused by the disguise.
This is the exact mechanism behind rheumatic fever. It's an autoimmune response triggered by a bacterial infection. The bacteria doesn't chew up the heart valves. Your own immune system does the damage.
That is deeply unsettling. Before we move on from the gram-positives, I want to talk about spores. The sources describe them as metabolic panic rooms.
That's a great way to think about them.
Yeah, some bacteria, when threatened, strip down their biology, pack their DNA into a core protected by dipicolinic acid, and basically go dormant. They are so tough you have to blast them with high-pressure steam at 121 degrees Celsius for 15 minutes just to crack them.
Spores are the ultimate survival mechanism. They can survive boiling water, harsh chemicals, and decades of environmental exposure without needing nutrients.
Which explains a huge mystery for me. The sources mention Bacillus cereus and reheated rice syndrome. Let's say I'm making dinner. I boil the rice to 212 degrees Fahrenheit. Every bacteria in there should be dead. But the notes say if I leave that rice sitting warm on the stove, I can get violently ill. How is that physically possible if I boiled it?
Because you kill the active bacteria, but you didn't kill the spores. They easily survive the boiling water. When the rice cools down and sits at a warm, comfortable temperature, the spores detect that the danger has passed.
Oh, no.
They germinate, transforming back into active, growing bacteria.
Yeah.
As they multiply in the warm rice, they pump out a preformed emetic toxin. You eat the leftover rice, and within 1 to 5 hours, you are violently ill.
So the spore is just waiting out the apocalypse. The text also mentions Clostridioides difficile, or C. diff, doing something similar in the human body.
C. diff is a vital example of how we inadvertently cause disease. It's an obligate anaerobe, meaning it only thrives in environments devoid of oxygen, like your lower intestine. Normally, it exists in low numbers, kept entirely in check by the billions of healthy, normal bacteria crowding it out for food and space.
But then a patient goes to the hospital and gets put on broad-spectrum antibiotics.
The antibiotics act like a forest fire, wiping out the massive ecosystem of good gut flora. But C. diff, tucked safely inside its spore panic room, survives the purge.
So once the antibiotics course through, C. diff wakes up to an empty ecosystem with basically no competition.
It germinates, rapidly takes over the entire gut, and releases toxins that physically destroy the intestinal lining. It causes severe water diarrhea and pseudomembranous colitis. The disease is a direct consequence of our own medical intervention, altering the microbial landscape.
Okay, so if the gram-positives are these heavily armored tanks and survivalists, let's look at the gram-negatives, the pink raincoats. These feel more like tactical guerrillas hiding in our food and water.
They rely much more on stealth and environment, yes.
The classic GI battleground is between Salmonella and Shigella. Both cause serious gastrointestinal disease, but their tactics are wildly different. Salmonella has flagella, little tails that let it swim, but the sources say if I ingest Salmonella, I need to swallow a massive dose, hundreds of thousands of bacteria to actually get sick. Why does it take such a massive invading army?
Because Salmonella is acid-label. It is highly sensitive to acid. When you swallow it, it plunges straight into the vat of hydrochloric acid that is your stomach.
Right.
The vast majority of the invading force is instantly dissolved. You need a massive initial dose just to ensure a few survivors make it through to the intestines to start an infection.
So what does this all mean for Shigella? Why does it need such a tiny invading army compared to Salmonella?
I think the note said it only takes 10 to 100 microscopic organisms to cause severe dysentery.
Shigella is acid stable. It is completely unfazed by the extreme low pH of stomach acid. It essentially swims right through the acid bath unharmed, meaning nearly 100% of the bacteria you swallow reach the lower GI tract.
And once there, it doesn't just sit on the surface. It invades the intestinal cells directly, spreading laterally from cell to cell without ever re-entering the open space where immune cells could spot it.
So a single trait, whether you can survive an acid bath, completely dictates the infectious dose. And these gram-negatives aren't just in our food, they are hiding in our buildings. Take Legionella pneumophila.
Ah, Legionnaire's disease.
Yeah, it hides in air conditioning units and hot water tanks. It's incredibly hard to spot on a normal stain. You need a special silver stain. I even read this bizarre mnemonic in the notes. Think of a French legionnaire with his silver helmet, sitting around a charcoal campfire with his iron dagger, missing his sister Sistine.
I know that one. That mnemonic is actually a lifeline for medical students trying to remember the incredibly specific laboratory requirements to grow it. It needs a buffered charcoal yeast extract agar, supplemented with iron and the amino acid cysteine.
Right, but the reason it hides in AC units is what makes it so fascinating. Legionella evolved to live inside microscopic amoebas in freshwater ponds, right?
Yes. And when we build complex cooling towers and warm water systems, we accidentally create the perfect artificial ponds.
And then the AC unit aerosolizes the water.
Exactly. It sprays microscopic bacteria-laden water droplets into the air. We breathe them directly deep into our lungs, bypassing all the normal throat defenses, resulting in Legionnaire's disease, which is a severe pneumonia.
Speaking of water, there's Pseudomonas aeruginosa. This one is described as smelling like grapes and producing a blue-green pigment. But it targets a very specific demographic: burn victims and people with cystic fibrosis. Why them?
Pseudomonas is the ultimate opportunist. It struggles to break through intact human skin or a fully functioning immune system. But when a burn victim loses that physical skin barrier, Pseudomonas thrives.
And what about cystic fibrosis?
In cystic fibrosis patients, the genetic disease causes a buildup of thick, sticky mucus in the lungs. Pseudomonas enters that mucus and produces a mucoid polysaccharide capsule, allowing millions of bacteria to stick together and form a dense biofilm.
So they build a fortified slime city in the lungs that antibiotics can't penetrate. We've mapped the purple tanks and the pink gorillas. But before we wrap up, we have to talk about the outliers, the rule breakers.
There are always exceptions in biology.
Always. Let's start with bugs that just refuse to take a gram stain, like Helicobacter pylori. It causes stomach ulcers. But wait, we just talked about the stomach being a vat of hydrochloric acid that dissolves salmonella. Here's where it gets really interesting. H. pylori surviving in stomach acid is like wearing a spacesuit in a volcano. How does it do it?
It uses chemistry. H. pylori secretes a specific enzyme called urease. Urease takes urea, which is naturally present in the stomach, and chemically breaks it down into carbon dioxide and ammonia.
And ammonia is highly basic, right? It's alkaline.
Exactly. So the bacteria isn't technically surviving the acid directly. It uses the ammonia to create a localized, neutralizing alkaline bubble around itself. It wears a microscopic antacid shield, allowing it to burrow into the stomach lining completely protected.
That is brilliant chemical warfare. What about the bacteria behind leprosy, Mycobacterium leprae?
Right, it refuses the gram stain because if cell wall is packed with waxy lipids, specifically mycolic acids, you have to use a specialized acid-fast stain. But the most defining characteristic of the leprosy bacteria is its temperature preference.
The sources say it prefers cold environments. But human core body temperature is 98.6 degrees Fahrenheit. Where does it go?
Well, because it cannot survive in the warm core of the torso, it is forced to migrate outward. It infects the superficial nerves and skin of the coldest extremities, the fingers, toes, ears, and nose.
Oh, wow. So that's why.
Yes, this is why leprosy patients experience nerve damage and loss of sensation, specifically in those peripheral areas. It's also why its primary animal reservoir in the United States is the armadillo, an animal with a naturally low body core temperature.
So if you step back and look at the sheer scale of what we've covered, I mean, from the way you must immediately put your leftover rice in the fridge, to why a burn victim is so vulnerable to a bug that smells like grapes, we've explored the structural armor, the jumping genes, the neurotoxins that lock our jaws, and the alkaline bubbles that let bacteria live in volcanic stomach acid. You, the listener, are constantly navigating an environment teeming with microscopic organisms that have spent millions of years perfecting the art of infiltration.
And that leads to a rather unsettling concept, something for you to mull over long after this discussion ends. Think back to how bacteria trade genes, how they use conjugation and transposition to pass around plasmids containing antibiotic resistance.
Right.
Consider your own gut microbiome, those 100 billion harmless bacteria in every centimeter of your colon. Over your lifetime, every time you take an antibiotic, you expose that massive population to the drug.
Oh, I see where this is going.
What happens if your harmless microbiome silently collects and hoards a massive library of antibiotic resistance genes? They act as a hidden vault, a microscopic sleeper cell, just waiting for the day they can hand that superimmunity blueprint over to a truly deadly pathogen passing through your system.
That is a chilling thought. We are carrying around the blueprints to our own destruction. Well, thank you for joining us on this exploration of the invisible world. Stay curious, stay informed, and maybe go check on that rice you left on the counter.